Hello, and welcome to Xtalks Spotlight, illuminating insights from subject matter experts and industry thought leaders. I'm Sonia Hunt. In this episode, we are asking the question, what challenges is the vaccine industry facing when dealing with the flu and COVID-19? With more, here's Aisha Rashid.
Thank you, Sonia. This time of the year, the U.S. population is gearing up for the fall and winter flu season. We've also been dealing with the ever-changing environment associated with COVID-19. Will we all need to get flu and COVID-19 vaccines this fall? The viruses that cause the flu, influenza, and COVID-19, SARS-CoV-2, have similarities and differences. Likewise, the recommendations for flu and COVID-19 vaccinations vary. This spotlight will help differentiate some of these concepts and provide information on the current situation with the COVID vaccine. We will also discuss what some of the challenges facing the vaccine industry for both COVID and flu are. For this discussion, we are delighted to have Dr. Lisa Kierstead from PPD, part of Thermo Fisher Scientific, with us in this spotlight feature. Dr. Kierstead joined PPD in 2009 and is currently an executive director of the Vaccines Sciences Lab. She has 23 years of experience working with clients to move vaccine candidates through the clinical development pipeline. This includes regulatory submission as well. She specializes in infectious disease and oncology, including dengue, norovirus, pneumococcus, meningococcus, and melanoma. Thank you for taking the time to join us in this spotlight interview, Dr. Kierstead.
Oh, thank you for having me, Aisha. It's nice to be here.
Thank you. So I'd like to start off by asking a fundamental question. How are the COVID-19 and flu viruses similar?
So there's several characteristics that make the two viruses similar and what they share. So the first one is their genetic material. So both flu and COVID have genetic material of RNA and not DNA. And what this does is it lends them to more mutation. And as we know, COVID is mutating all of the time, and we're seeing variants. And so an RNA genome enables the genetic material to mutate more rapidly. So that's a similar characteristic. Slight difference is that influenza is what we call a single-stranded negative sense RNA virus, and COVID-19 is a positive sense single-stranded RNA virus. What that means simply is COVID-19, once it gets into your cell, can right away replicate protein. And so its cycle of replication is faster than influenza. So that's a slight difference, but they're both RNA genomes. Secondly, they both infect multiple species, which is interesting. So flu can infect birds and pigs, and COVID-19 can infect cats, dogs, multiple species. So that's another characteristic that they share. And then finally, infection with flu or COVID both result in respiratory symptoms. So slightly different, but they both result in respiratory symptoms. So those are the similar characteristics that the viruses share.
And what is the current recommendation for flu and COVID-19 vaccinations?
So the current recommendation for flu, which has been established for quite a while, is an annual vaccination. And it is for anyone that is greater than 6 months of age, unless you have what they call a contraindication. So if you have an allergy to eggs, for example, that's considered a contraindication. And that vaccination is usually recommended in the September to October timeframe. Now, COVID, on the other hand, the current recommendation is similar in that everyone above the age of 6 months is recommended to receive the current authorized vaccine, which this year is a variant called Omicron XBB.1.5. So there are similar recommendations for flu and for COVID in that they're annual and similar ages as far as the recommendation.
And so speaking about these vaccines, how are the currently approved vaccines for flu and COVID-19 similar?
So they actually don't share very many similarities. So I'll start with flu, for example. So for flu, generally the vaccines are whole, inactivated, or attenuated viruses that are typically produced in egg or cell culture based manufacturing processes. And there are several manufacturers with clinical trials going on now to actually look at an mRNA based flu vaccine and seeking approval for that. So that's flu. In addition, flu is what we call quadrivalent. So there's actually 4 seasonal strains that are included in the flu vaccine that is recommended by the WHO. So COVID, on the other hand, as we know, right now the 2 approved vaccines for the fall season are both mRNA based vaccines that encode a specific protein, and that protein is called spike. And that is a monovalent vaccine. So it only has one genetic sequence, which is from the variant, as I mentioned, which is at XBB.1.5. So those are some similarities, but differences, mostly differences actually, between the flu and the COVID vaccine.
What are correlates of protection, and do they exist for flu and COVID 19?
So a correlate of protection or surrogate of protection, a high level definition of that is simply scientific data that correlates with protection from either infection or symptoms. And universal correlates of protection are challenging to establish. We do have some what we call legacy vaccines that do have correlates of protection. A couple examples are measles, which has been around for a very long time, and pneumococcus is another example. For measles, the correlate of protection is if you have a certain concentration of antibodies in your system, then you'll generally be protected from getting measles infection. Now, the challenges to correlates of protection is they don't always hold true. And the reason for that is when you think about the globe, right, there are different populations of individuals around the world. There are different ages of individuals around the world. And to complicate matters, for example, if you talk about pneumococcus, there are more than 90 different types of pneumococcus. So one correlate of protection, a single, value cannot hold true for everything, that there is. And so there's a lot of complexity around establishing correlates of protection. So as far as flu and COVID, there's publications for both actually that suggest for flu, for example, if you have antibodies to a certain protein, which is called hemagglutinin or HA, and you have a certain antibody level that is determined by a certain type of method, which is called a hemagglutination inhibition assay. If you have antibodies to a certain level, that you'll be protected from flu. That's, again, generally. It's not a universally accepted, there isn't a single value that's been established, but that method will, if you have those antibodies, you'll be protected. And for COVID, more recently, and COVID is a much more new, virus to us, what we call neutralizing antibodies, which those are essentially an antibody in your system that can neutralize the virus so it can't get inside your cells. So it's been shown that if you have a certain level of neutralizing antibodies to COVID, then you'll be protected. But again, it's not a defined universal criteria. It's just there seems to be a correlation between antibodies that function in a certain capacity and protection from flu and or COVID.
And speaking of complexities, what are some of the challenges facing the vaccine industry in dealing with flu and COVID-19?
So probably the most common, biggest challenge for both flu and for COVID-19 is because they're RNA viruses, they have a tendency to mutate. Flu doesn't mutate as quickly as SARS-CoV-2, which causes COVID. And we know COVID variants keep popping up and popping up. But to talk specifically about flu first, so every year, the seasonal flu strains are defined for the northern and the southern hemisphere. And those strains are selected, the strains for the vaccine are selected by WHO and obviously flu experts. Once the vaccine is selected, then the manufacturers will generate the vaccine with the relevant strains, and then it'll be given to the public. And sometimes the flu, so first challenge, sometimes the vaccine itself isn't effective against the circulating strains, right? So that's one of the challenges. The other challenge, and that could be the result of one or two things. One, perhaps they selected the wrong strain, to generate the vaccine to. That's one possibility. The other one is, even if they have selected the right strain, sometimes what we see is there's only a 50 to maybe 65% effectiveness of the flu vaccine in preventing infection. And so there's challenges there. And there's been a lot of discussion around that, of course. And some experts will say, well, the flu vaccines that we have, some of the production processes have been around for a long time, and perhaps they need to be reassessed and improved. So that's one example. And as I mentioned before, there are clinical studies currently going on where mRNA-based flu vaccines, which is a similar process to what was done for COVID, are in clinical trials. And so if those are approved, perhaps those will be more effective, and they're more readily produced, quicker to produce than some of the cell-based or egg-based flu vaccines. So there's some activity going on there, which would obviously potentially benefit the population. And then for COVID, I think the biggest challenge, as I mentioned, is the rate of mutation. So we continue to see new variants popping up. So to talk about COVID and the challenges associated with COVID vaccination is, again, it continually is mutating, and the variants are arising at a relatively quick pace. And so it causes a lot of challenges. And one of the things that I wanted to mention, though, although it is a challenge to the industry that the virus keeps mutating, with our September, October vaccination recommendation, which is for the Omicron XBB.1.5, that vaccination may be effective against variants that pop up. We just don't know, right? So just because we have variants popping up doesn't mean the vaccine won't be effective against those strains that pop up. All right. So we just don't know. So we have the variant that's been selected, and I think in the U.S. and abroad, the same virus has been selected. So we'll have to just see what happens with COVID. So.
Absolutely. And on the COVID front, what new progress has been made regarding COVID-19 vaccinations?
So I think, I was thinking about this, and one of the things that I've been really impressed by is the fact that the vaccine manufacturers have been so nimble in responding to the appearance of new variants of concern and being proactive, right? So even before the XBB variant was selected for the September, October vaccination, the manufacturers were being proactive and already running clinical studies with that variant to see if their vaccine was effective and whatnot. So that once that variant was selected, and that was in June, okay, just in June, they were able to get a vaccine to market in the September, October timeframe and authorized, which is astonishingly fast and very nimble. So they're taking a lot of risk upon themselves to do that work, prior to knowing that the selected vaccine variant was going to be XBB. So I think that's one of the things that I find extremely impressive. The other thing is that there is funding now from the Department of Health and Human Services for what they're calling next generation COVID vaccines, and specifically looking at novel approaches, different approaches than what we've already seen from a vaccine perspective for COVID. So that is also a lot of progress there as well. So I would say, in my mind, those are the two things that I would say there's been a lot of good progress as far as COVID-19 vaccinations go.
Wonderful. Dr. Kierstead, thank you so much for speaking with us today in this Spotlight feature.
Thank you. I really appreciated your time, and I hope you have a great day.
Likewise, thank you once again. We look forward to keeping our audiences updated on the latest developments in flu and COVID-19 vaccinations. Thank you all for joining us for this Xtalks Spotlight feature. We hope you enjoyed the discussion.